Hello, I'm Joe Takahashi at the University of Texas Southwestern and the Howard Hughes Medical Institute. And for this second lecture, what I'd like to do is to give you 3 examples of using genetics in the mouse to really discover genes that are involved in controlling circadian behavior. And so the way a genetic screen in a mouse is performed is to use a chemical mutagen such as ENU, or ethyl nitrosourea, which is used to treat a male mouse. That causes mutations in the germline of that mouse. And then that treated mouse is crossed to normal female mice in what we call the G0 generation. And these mice then produce a number of offspring that we call G1 or generation 1 mice. These mice are carrying mutations that they inherit from this treated mouse up here. In a recessive screen, which is a little bit complicated because it requires 3 more generations of crossing, a male mouse is then crossed to another normal female mouse to produce a second generation of progeny. And then the female progeny in this second generation, or G2 generation, are actually crossed back to the G1 male. And that produces the third generation of mice, which are called G3s. And the reason we do this is to make any mutation that was heterozygous or single copy in the G1 mouse homozygous. And in this particular procedure, we try to screen about 20 G3 progeny for each G1 mouse. That gives us about an 85% chance of detecting a homozygous mutant mouse in the G3 generation. So in this kind of screen, shown here where we use circadian rhythms, this particular screen we screened over 3,200 mice. And this histogram here shows you the distribution of period values, similar to what I discussed in the first lecture, for these 3,000 mice. What's remarkable about this behavior is how similar each of the mice are to each other. So the average in this experiment was about 23.2 hours for the whole population of 3,000 mice, but the variation within that population was only 0.2 of an hour. That's only 12 minutes between mice. And so in this screen, this is, again, an activity record of a wild type or typically normal mouse, first on a light cycle, then transferred to darkness so we can see its circadian period. We found this mouse, shown in the middle panel, which has a 26-hour clock. And this mutant we named Overtime. It turned out that this mouse didn't breed, but we were able to recover the mutation from a SIB, this mouse below, which turned out to be a heterozygous carrier. And using this mouse, we were able to map the mutation to this region of the genome. And once we were able to reduce this interval, we found that there was only a single mutation in this gene here, which is called FBXL3, which is an F-box protein. Now, an F-box protein turns out to be involved in a complex of proteins that are involved in modifying other proteins with ubiquitin chains. And these ubiquitin chains then target that protein for degradation in the proteasome. So to make a long story short, FBXL3 uncovered a new F-box protein that targeted the cryptochrome protein for ubiquitination, and that mark then tags cryptochrome for degradation. And in the absence of this enzyme, or loss of function of FBXL3, the degradation of cryptochrome protein is slowed down tremendously. And what that does is to prolong the circadian period and lengthen the period from 24 to 26 hours. So we can see that this particular step accounts for a few hours out of 24 in the feedback circle. So in a second screen shown here using a slightly different strain of mouse, so the period of the rhythm is a little longer. It's 23.7 hours in this case. We found a short mutant, and that's shown here. It's only subtly short, about a half an hour short. Named that mutation pastime. And when we mapped this gene, it turned out to be in a new position. And incredibly, this gene turned out to be another F-box protein. This time, it was called FBXL21, which is called the paralog of FBXL3. So a paralog is a gene that has a very similar sequence that arose from gene duplication in the same organism. And so we assumed that FBXL21 should have a related function to FBXL3. But the mutant is surprising because it has an opposite phenotype. Instead of lengthening the period, as we saw for overtime, this mutant shortens. And so we looked for the interaction of overtime, the long period mutant, with pastime, the short period mutant, by crossing those two strains of mice. And the bottom shows the double mutant, and these histograms show the average period or the period distribution of these four genotypes of mice: wild type, overtime mutant, which are 26 hours long, pastime mutants, which are half an hour short, and then the double mutant, overtime and pastime. And what you can see is that the pastime mutant actually neutralizes the period lengthening effect of overtime and normalizes the period. So this kind of genetic interaction is non-additive and is interesting because it suggested that pastime was somehow counterbalancing the effect of overtime. Now, I'm not going to go into all the details, but I'll just jump to the end of this story, and that was that we found, to our surprise, that FBXL3 is really only found in the nucleus, but FBXL21 is found in both the nucleus and in the cytoplasm. And that in the nucleus, there's a very fine balance between FBXL3 and 21 interaction for the cryptochrome protein. And what happens is that FBXL3 is normally promoting the degradation of cryptochrome, but 21 actually competes for that and protects cryptochrome from degradation. And so that's why we think the two genes have opposite effects. 21 protects cry. FBXL3 promotes cry degradation in the nucleus. So they're counterbalancing there. But the surprise is in the cytoplasm, FBXL3 still degrades cry, but it does more weakly than FBXL21 still degrades cry, but it does so more weakly than FBXL3. So the discovery of these two mutations in related F-box proteins has, first of all, told us that the nucleus is very important in controlling the degradation of cry, and that the primary effect of these mutations on period length appears to act at the level of the nucleus, not the cytoplasm. So that was a little bit surprising. And the second is that related F-box proteins might have very different functions in the cell, both location, but also on their effects on the clock system even though they look very similar at the sequence level. Okay, so this has really led to a revision of our clock gene network shown here. This is the core clock BMAL per cry feedback loop. And now we've modified the diagram so that FBXL21 is really the primary E3 ligase in the cytoplasm. There is no FBXL3 as we thought before in the cytoplasm. Instead, most of the action is actually occurring in the nucleus, where FBXL21 and 3 are competing for cry in a very fine balance to regulate the stability of the cryptochrome protein.